Okay, so I want to start talking about meiosis and genetic diversity just a little bit. So to start off, I'm going to talk about meiosis 1. Meiosis, and I'll use the Roman numeral 1 here. There is, if you remember, 2 divisions of meiosis, and we'll go over that in the next slide, but let's do the first round of division for meiosis right here. First thing I'm going to do is I'm going to draw myself a big nucleus here. I'm drawing it big because I don't have too much room in these circles here. So let's draw a nucleus, and I'm going to simulate interphase right here, and I'm going to draw a couple of chromosomes like this, and I'm going to draw a couple of chromosomes like this. You'll notice I drew 4 total chromosomes. This is a diploid cell. So you might be asking yourself, why did I draw a red one and a blue one? I hope that'll be obvious, but there is one from dad and one from mom, right? Now what's going to occur during this process of interphase right here, and this is interphase, I'll just put an I can write it out, interphase, right? An S phase of interphase. Even in meiosis and mitosis, you're going to have these chromosomes kind of duplicate. Right, and by the time we get to prophase over here, we're going to have duplicated DNA, and you're going to end up with not much of a nucleus left over. Let me switch back to red here. The nucleus is probably pretty much broken down. You get your centrioles forming, and you should end up with something like this. Let me get back to blue over here. Now you have duplicated DNA, right? You have these chromosomes. Each of these chromosomes has individual sister chromatids with identical DNA at this point. Now, another thing that happens in prophase 1, I'll put a 1 here to indicate that this is prophase 1, is you're going to have, and I'm going to erase this over here, you're going to have some of these chromosomes get together, and they're going to combine next to each other, right? And they're going to swap out DNA over here. They're going to kind of swap pieces of DNA. And you're going to see that happen all across this cell with all homologous chromosomes, right? These chromosomes that are meant for each other, that have the similar genes, but they're from different parents, and they're going to swap DNA at this point, right? So what's going to happen is we're going to move into metaphase 1, and these chromosomes, due to this, and I'll write it up here, the forming of these synapses, right, where DNA is swapped. This is known more commonly as crossing over or recombination, genetic recombination. But that occurs right over here. So let me go ahead and switch inks one more time. Now we don't have a nucleus anymore now, but we are going to have some chromosomes that have swapped DNA, and they're starting to line up here in the middle a little bit differently, right? And I'll try and indicate that these are swapped by this right here. Let me get these closer. There we go. You'll see where I'm going with this in a sec, hopefully. So now you'll notice these chromosomes aren't identical anymore already, right? Let me do this one out down here as well. I'll just do like that, and like that. Go back. Whoops. Okay. So we have these very genetically unique now chromosomes lining up along this metaphase plate here, only they're lining up a little bit differently than what we saw in mitosis. If you'll notice, the homologous chromosomes are lining up next to each other, right? Rather than, if you'll remember, this is I'm going to draw a very brief round of mitosis over here to show you guys how it would look were this mitosis. You would have something like, let me get the blue out again here. You'd have them lining up something like this, right? And there would these sister chromatids would be ready to come apart. In this scenario in meiosis so far, these sister chromatids are not going to come apart. These chromosomes themselves are going to separate and segregate into separate halves of the cell. So, interphase, on to prophase 1, where crossing over occurs. Then you move on to metaphase 1, where you have these homologous chromosomes lining up right next to each other along this metaphase plate. And then, as you know, next comes anaphase 1. And anaphase 1, you're going to have these, as you might guess, these chromosomes are going to pull into opposite sides of the cell. So in the interest of time, I'm just going to draw these as quickly as I can here. Let's see. So, end up with kind of something that looks like this. Right? These are going to start pulling towards these opposite poles of the cell. Now, you're going to end up with, in telophase, and I guess I should have drawn this sideways, but we can pretend, right? You're going to end up with these pulling over to both sides, and I'm just going to flip this cell to the right a little bit just to make it look a little bit better the way this elongation is working here. And we'll end up with that. And then over here we'll have something like that. Okay. Right, so we have something like this pulling over to both sides. And of course this is telophase 1. Telophase 1, and then of course this ends up with cytokinesis, right? And so you should eventually end up with these two cells that each are, at this point, haploid cells. Haploid cells. After one division in meiosis, you have haploid cells. So you'll notice, let's see, you're going to end up with, I'll just draw these down here again. Sorry for the tedium. Maybe I'll pause this next time when I do this. I probably will have to. Go ahead and draw this in your notes if you would. If you have different colors, it's even better because it shows where this DNA came from originally. Okay, so now we end up with these two haploid cells. We're done with meiosis 1, but we're not finished with everything yet. You're going to have, of course, I forgot to draw this on here, but you're going to have a nuclear envelope briefly reform in telophase. Of course, in here you'll have, kind of a nuclear envelope. Of course, this nuclear envelope is getting ready to break again, as we're going to see. But let's go ahead and move on to our next phase. Make sure you're with me so far. Interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. All right, let's move into meiosis 2. Meiosis 2. Right? So what I'm going to do is I'm going to take just one of these cells over here, just to save time. I'm going to take this cell right here, and we're going to follow this cell along its path, right? Onto this, into this next phase. So let's go ahead and follow this cell. This cell right here. I'm going to kind of redraw it up here. Right, you have your, of course you'll notice here we don't go back into interphase, right? We just kind of move straight on to the next step. Right? And what we're going to have happen so now is we're going to move, there is no real interphase to speak of, right? So we're going to move straight into, and I'll draw, and we'll call this prophase. Prophase 2 right here, right? And we'll just skip this guy and go straight over here. Prophase 2, we don't really have an interphase in this one. So, prophase 2, of course you'll know, please make another touchdown. PMAT. Next one we're going to talk about is metaphase. Metaphase 2 here. And now you're going to notice that these things line up a little bit more like you're used to, like you saw them in mitosis. So they're going to line up kind of like they did mitosis, and now you're going to see these sister chromatids. Of course, they're not really sisters anymore, are they? They're kind of genetically unique at this point, right? So these are going to line up again, and you're going to see in anaphase 2, you're going to notice these things kind of pull to separate sides of the cell. Oops, I should have stayed here with blue. Okay. Make this one longer. This is the little tiny one over here. This one's longer. Okay. Oh, I drew those the wrong way. These should be facing the other way, but regardless, you see what you get the point I'm making. These both move towards opposite poles of the cell. And of course, eventually you end up with one cell is going to look kind of like this, and the other one's going to look something like this, right? And this is telophase 2. Telophase 2. And then eventually, of course, you end up with cytokinesis occurring. Okay, cytokinesis has occurred here, and you end up with 2 genetically unique cells. Of course, keep in mind, there would be, out of this, 2 more cells over here. I didn't generate those, but they come from this other cell, which would also divide, right? This would give us 2 other genetically unique cells up here, get for a total of 4 genetically unique cells. So I would like you to go ahead and take that other cell on your own and kind of draw out its path for meiosis and see how that goes. It should do it exactly the same as I did it over here. Of course, try and keep your colors straight and kind of follow this through the process and end up with 4 total cells. I'll let you guys figure that one out. But in the end, you will have 4 genetically unique cells. Now you might be asking yourself, where does this genetic diversity come from? Why do we get genetically unique cells? And there's 2 reasons for that. One of those reasons is this crossing over example up here. You'll notice they get next to each other, they cross, they swap DNA, right? Typically closer to the ends of these chromosomes. It's going to happen much less towards the middle. And then another reason you have genetically unique cells is once these things do line up in metaphase 1, they can line up with either one of these, 1 or 2, 1, 2. They can line up like I have them here, or these could have been flipped, and you could have had this chromosome over here and this one over here. They can arrange in many different ways. They can assort themselves in many different ways. So, this is kind of the basics of meiosis and how it works and how you end up with 4 genetically unique cells. I want to overview the whole process here. You might want to pause the video and take a look at this so you can follow it in probably a better graphic than I can do with my pen over here. But you'll notice that you have crossing over in prophase 1. The metaphase 1, they line up along this metaphase plate, but they line up right next to each other. These chromosomes line up next to each other. And then the homologous chromosomes separate from each other. Sister chromatids, of course, remain intact. Of course, it's hard to call them sisters at this point because look at them. They're already unique. Then you have that division. Prophase 2, you have a similar process occurring, only this time it looks a lot more like metaphase for mitosis. Of course, from this point on, you have haploid cells, right? Haploid cells after that first division. And they will remain haploid for the rest of their life until you end up with N down here at the bottom, 4 genetically unique haploid cells. Now, what's nice about this is if you have one sperm over here, and one of these is an egg, of course, a different organism would have to make an egg, but obviously. But this would give you the full complement of chromosomes if you were to combine these two sets of DNA right here. You would get one total human being, right? And that's why you are who you are. It's why you're different from your sister, parents, and other people around you, is because of this process, which creates a massive amount of genetic diversity. So that's the basics of meiosis. I will see you in the next video. Thanks.